to our final presentation in this auditorium. And I think it is a fitting end to at least the weekend up here. Mark Painter will be presenting on immunity, COVID-19 and beyond. This presentation is a scientific presentation on how our immune system responds to viruses and vaccines. Mark, who's with us this afternoon, is a post-doctoral fellow at the University of Pennsylvania's Perelman School of Medicine, specifically in their Institute of Immunology. So let's all welcome Mark. Thank you.
All right, thanks Rebecca, and thanks everyone for being here. I'm excited to hopefully share with you a little bit about what I think is the coolest thing in the whole world, which is the immune system and how it works. And I think before we get started, I just think it's really important now more than ever. I think we're all used to people when it comes to vaccines and the immune system telling us what we should do. Don't get the vaccine, get the vaccine, and often not giving all of the reasons for why they think what they think. So I think today instead, the goal is going to be hopefully to share what we know about how the immune system works. I think it's marvelous, so hopefully you'll think it's fun, but also hopefully to equip you to think for yourselves, to be free, and to really know why you're doing the things you're doing instead of just being stuck listening to whoever's loudest. So to start, I think we need to think about what is a living thing, and we're going to be spending a lot of time here during this presentation inside the cell. So I just want to sort of orient you to how life unfolds within a cell in the normal context of a living thing. So we're going to be thinking mostly about a human immune response. So inside a normal human cell, we have information encoded as DNA, right? DNA is inside the nucleus. So here's a cell, you have a cell membrane, there are things outside the cell up here. And then inside the cell you have the nucleus, and in the nucleus you have the DNA. We can think of the DNA as like a precious original copy instruction book that has instructions for how to build everything in a cathedral, right? The cathedral of our body, right? So it has the windows, it has the arches, it has the chairs, the pews, the podium, everything that's going to be in the cathedral, right? But we only have one copy of the original instructions, so it's very precious. We protect it inside the DNA. If we actually want to build that cathedral, build the cell, build the human body, we make photocopies of the information that's in that precious book in the DNA. And those photocopies are called messenger RNA, or mRNA, which I think we're all familiar with the phrase mRNA now, thanks to the remarkable vaccines that were developed, right? So this is what an mRNA is. It's a photocopy with the instructions for how to build something. And what does it build? It provides the instructions to build proteins. So from the DNA, we make a photocopy, it's called transcription, of an mRNA that leaves the nucleus where it encounters these ribosomes that just read whatever instructions you give them. If you give them instructions, they'll read them and they'll build. They're great builders, and they build the protein that was encoded in the DNA, right? Whoops. The protein that was encoded in the DNA. Right? So this is how we build everything. Proteins are what do everything in our cells. They're what almost everything. They're what make everything that you see, all of the activities. So you can metabolize, you can take up nutrients, you can build a heart, you can have eyes that see, your brain can work. This is all done through building specifically the proteins that you need to live, right? So this is the flow of normal information in your cell and the cells in your body. Every cell in your body is doing this all the time. And producing the proteins that make you what you are and make life what it is. So in this context, what is a virus, right? And a virus really is a very simple organism. It's made up of just a little piece of genetic information. It could be DNA, it could be RNA that hijacks this system, right? And I think it's helpful to understand exactly what it is that a virus does, right? So in the normal flow of information in our cell, you have your DNA. You're activating the right genes, making your photocopies of mRNA to build the proteins you need to survive. When a virus comes into your cell, it's floating around outside the cell, and it sneaks in through a receptor and gets inside the cell. And this is a diagram I made of SARS-CoV-2, right? So this is the virus that causes COVID-19. This virus, by chance, it happens to be an RNA virus. So the viral genome is a little piece of RNA that instead of providing the instructions for how to build the things your body needs, it has the instructions for how to build more virus, right? It inserts itself in, and your ribosomes, right, good workers that they are, simply receive the instructions that they have and start building proteins. You can see the problem here, right? So the things that you need to survive are now working for the virus. And what they build are all of the proteins that the virus needs to form new virus particles, leave the cell, and go infect the other cells in your body, and go infect other people who you come in contact with in your life, right? So in the case of SARS-CoV-2, it's an RNA virus. It has 14 genes. It's very small. We have over 10,000 genes, more or less. This virus only has 14, and that's all it needs. If it can provide the instructions to build these 14 proteins, it can package up another little piece of this RNA and go and continue the infection. The problem is that once this cell is infected, all it will be able to do is produce new viruses, right? This cell can no longer, because it's flooded with this information from the virus, it's unable to keep functioning as a normal, healthy cell. And this is a big part of what makes you sick. Right? Just to tell you, like, viruses can take different strategies. So I don't want to just tell you about COVID. I want to help us understand the immune system in general. So this is HIV. This is the virus I studied in graduate school before COVID happened. And HIV is a little different, but you'll see that ultimately the process is the same. So HIV is clever. It actually comes in as a piece of RNA, converts that RNA into DNA, and then sticks its own DNA into your DNA. Very, very sneaky. And then from there, it can produce its own mRNA molecules that provide the instructions to build the proteins to build HIV viruses that can then leave and go infect another cell, right? But you can see that what a virus really is, right, is just a little piece of genetic information that hijacks the normal pathways in your cell, takes over your cell that was doing what you needed it to do to survive, and instead turns it into a virus factory, right? And that viruses can achieve this by all different ways, right? You can have a virus like SARS-CoV-2, you can have a virus like HIV, but ultimately they work with the same principles. Providing some genetic information, some mRNA that encodes instructions for building virus instead of building you. So the question that we have, and the focus of this talk, is going to be how does our immune system actually protect us from viruses? And we're going to talk, because I love immune cells, we're going to talk about two different kinds of cells, B cells and T cells. So B cells are called B cells because they're made in the bone marrow, or they're educated in the bone marrow. And this is what B cells do. And we'll talk more about B cells in a minute. But what B cells do, to be overly simplistic a little bit, is in a normal situation, if you didn't have any B cells, or before you've generated any immunity, the virus can come in, right? It's floating around, it can bind to those receptors on the cell, and it can get in, and it can infect that cell. And then that cell becomes a virus factory. Bad news for you. If you have functional B cells make antibodies. I think we've all heard of antibodies before. They're one of our favorite things to talk about these days. And B cells are the cells that make antibodies. And the antibodies, what they can do lots of things, but the thing that we're going to focus on them doing mostly today is they can neutralize the virus. So they can, with high specificity, stick to those parts of the virus and block it from ever getting into the cell. So if you have enough antibodies, if you have B cells that have made antibodies that can protect you against this virus, you might never get infected in the first place, because the virus never can gain entry into the cell. As soon as it gets close, it gets neutralized, it never gets in, and this cell gets to carry on doing the things it's supposed to do instead of becoming a factory for virus. Those are the B cells. The T cells work a little bit differently. T cells are called T cells because they're educated in the thymus. So B for bone marrow, T for thymus. And what T cells do is they say, okay, let's say our antibodies failed, and we do have cells that got infected. Those cells have become virus factories. They're no longer useful to us. In fact, they're quite harmful to us. So what we really need to do is get rid of those cells. And what these T cells can do is they scan along and they sort of check in with each cell in your body. And they ask, are you normal or do you have something wrong with you? And if a cell has something wrong with it, namely in this case that it's infected with a virus, it'll put up this little signal, hold a little piece of the virus out to the T cell for the T cell to see. And the T cell says, okay, that's bad news. This cell is infected with a virus and it will kill this cell. brutal. But this cell was only going to be a virus factory from this point on, right? So really the best thing you can do is get rid of it. And they'll scan along and they'll check every cell, and if a cell is infected with a virus, you can kill it. And so you can see how these two mechanisms are both valuable, right? If you have antibodies, you may never get infected in the first place, and you may be able to stop the spread of the virus within your body by preventing it from entering new cells and turning more cells into virus factories, right? But if you did get infected, you need something to clear up those cells so they'll stop producing virus, and eventually you can actually get over the infection. So we have both B cells and T cells which do these things. But I think this opens a big question, which is how the heck does a B cell or a T cell know how to recognize SARS-CoV-2 or COVID-19, right? The virus that causes COVID-19. How on earth could it possibly know? And this, I think, is the absolute coolest thing in the world. I'm going to leave this slide up for a bit, and we'll talk about how B cells and T cells work. So when you generate a new B cell or a new T cell, B cells you generate throughout your life. You'll generate billions, tens of billions of them during your life. Every time you generate a new B cell, it has to decide what is the one antibody that it is going to make, right? So each B cell can only make one kind of antibody. It might be an antibody for COVID-19, it might be an antibody for flu, it might be an antibody specifically for the Omicron variant, it might be an antibody that doesn't recognize anything that exists yet in the world, which is what we would have said about our COVID-19 antibodies 4 years ago, right? But it has to decide what kind of antibody it's going to make. And what it does is, in your DNA, you basically have 3 piles of Legos, right? A pile of red Legos, a pile of green Legos, and a pile of yellow Legos. And when a new B cell is born, what it does is it randomly picks one of the red Legos, one of the green Legos, one of the yellow Legos, and puts them together. And it puts them together in a random way, right? Think of all the different ways you could attach 3 unique Legos, right? And that is the antibody that it makes, right? And antibodies work by sticking to something very specifically, right? So if you think about 3 Legos, only a very precise piece could fit it. But if the virus had that piece, it would be a perfect fit, right? So you generate tens of billions of B cells, each with their own antibody, each one totally unique, through this mechanism of randomly generating something, with no idea of what it might recognize, right? And T cells do the same thing. So the T cell receptor, so B cells make antibodies. Before the antibodies made, it exists as the B cell receptor, but it's the same molecule. T cells do the same exact process with the T cell receptor. So they take one red Lego, one green Lego, one yellow Lego. They stitch them together in a random way, right? And so through this, you generate a tremendous diversity of T cells and B cells, an unbelievable diversity. In fact, like an almost infinite diversity that will be capable of recognizing virtually anything that could ever exist and could ever enter your body, which I think is really remarkable. And I hope it's clear how this works. So it's totally random. You generate all of these random cells, and they go out. They have no idea what they're going to do, right? I hope you may already be thinking, right, well, this could be problematic, right? Like if it's totally random, like what if I make a B cell or a T cell that recognizes, my eyeball, something that's in my eyeball, right? And it goes out and starts killing my eyeball. That would be very bad, right? So when I mentioned before that these cells get educated, they're generated in the bone marrow, but then they receive an education. And the education is also a brutal one. Hate to say it, immunology is not always a nice place to be a new B cell. So when a new B cell or T cell is made, here we have 7. We made 7 different random combinations. You can see this nice little picture from Janeway's immunobiology textbook of how they have a slightly different shape. Right? Now imagine of these 7 represent really billions of unique T cells or B cells. They then receive this education where in the thymus and in the bone marrow, there are special cells that actually express every single gene in your body, right? And so they show every new B cell and every new T cell that's made, everything that you are. The genes in your eyeball, the genes in your brain, the genes in your toenail, the genes everywhere, right? The genes all throughout your body. Right? And if any of those B cells or T cells recognize any of the proteins that your body is supposed to make, they kill themselves, which, sad, but it's because they're dangerous, right? And if you have a failure at this step, you can get a disease like autoimmunity, right? Where your own cells start to attack your body, like you see in type 1 diabetes, where you have cells that go and attack the insulin-producing cells in your pancreas, right? It's a failure in this step of immunity. But luckily, in most people, this works great, right? And the B cells that leave the bone marrow and the T cells that leave the thymus have received an education that says these cells are not dangerous to me. They very well may not recognize anything that actually exists in the world, but they might, right? And they leave, and they go out and they hang out in your lymph nodes, in your spleen, they circulate in your blood, and they wait. And actually, at this stage, they can't do anything. They're called naive cells. A naive T cell can't kill anything. A naive B cell can't produce its antibodies, right? And they wait there, and they wait and they wait. And most of them, most likely, don't actually recognize anything that exists. And they'll wait your whole life and do nothing. They'll sit there and do nothing. They'll circulate through your blood, wondering if they'll ever find, right, the thing that fits perfectly, what they were made to fit, right? And they probably never will. But when something new comes along, when a new virus comes along, right, those cells that have been waiting your whole life will come into contact with the piece of that virus that fits perfectly with their antibody or with their T cell receptor. And this sets off an explosion of life for that cell. So that single naive cell that was sitting there waiting, unable to do anything, suddenly starts to grow. It gets bigger. It starts to divide. It produces thousands, millions of copies of itself because now it knows I'm a useful cell, right? I'm among all these B cells that make antibodies that don't recognize anything, my antibody matters, right? Because it recognizes something that is actually, that I'm being exposed to right now. So we want more of me, right? So you make tons of copies of these cells that then can produce tons of antibodies if they're a B cell. Or if they're a T cell, they can receive a license to kill, right? And they can actually start to go out and kill whatever it is that they recognize because they've been activated. So that's what's represented here. That when the foreign antigen, fancy immunology word, for the thing that sticks to a T cell or a B cell, is recognized, it sets off this huge expansion and production of antibodies and T cells that can go out and actually get rid of that virus. So that's how T cells and B cells work. I hope that's clear. So you understand that the B cells then, you generate tons of them, and when the virus comes, they can actually start to produce antibodies that can prevent infection. And the T cells, you generate tons of them, and when the virus comes, you can actually start to make T cells that actually are capable of killing things. You can make millions of them that can go out and actually kill all the virus-infected cells to get rid of the infection. So now we're going to talk about how that actually happens, right? So you probably have some idea already. There's actually two kinds of T cells. I may not linger on that too much. But you have your naive B cell and you have your naive T cells, right? If the virus shows up, the naive B cell, like I said, there's an explosion of life. There's also a diversity, right? They can take on different forms. They become capable of things they couldn't do before. So this naive B cell can become a special cell called a plasmablast, which doesn't live for very long, but what it does is it produces a ton of antibodies as fast as it can, right? So this naive B cell makes a bunch of antibodies that can go out and neutralize that virus that came in and infected you. It can also, and this is really important, become two other types of cells, right? Through help from T cells, it can become a long-lived plasma cell. These are kind of the gold mine, because they go hang out in your bone marrow and produce antibodies for as long as they live. And some of them live for the rest of your life, right? So these short-lived plasmablasts produce a bunch of antibodies, and those antibodies, they degrade over time, right? They're just proteins floating in the blood. So every 30 days or so, their levels will drop by half. So over time, those antibodies that are produced in this initial wave to try to get rid of the virus as fast as possible will go away. You may generate some of these long-lived plasma cells that will keep producing antibodies and keep you protected from infection for the rest of your life, right? But you also make this important pool of memory B cells that instead of a single naive B cell, you generate these memory B cells that have more capacity. They're ready. They're like twitching, ready to make antibodies at a moment's notice, right? If they get any sense that virus is back, they'll immediately produce a ton of antibodies. And the cool thing is that when you generate memory B cells, they also diversify a little bit. They make little mutations along the way. So these antibodies, let's say that these B cells that make an antibody that recognizes COVID-19, the original one, right? They'll get a little bit different. Not much, but they'll make a couple mutations here and there. So that if the virus ever changes ever so slightly, right, you actually have a pool of memory B cells ready to make antibodies that might be even better at fighting off that virus, which I think is totally amazing, you know, because viruses do change, and so it's really amazing that we've evolved this mechanism to do this. On the T cell side, it's very similar, right? So your naive T cell can't actually do anything. It encounters the virus, and you make a ton of those T cells, because you have to get rid of this virus. So you make a bunch of them. They go out as effectors, we call them, and they actually eliminate all of these virus-infected cells to slow down how much you transmit the virus to other people, to actually clear the infection so you can go back to living your healthy life. Most of those, once the virus is gone, will die, because you don't need millions of T cells running around looking to kill something that is no longer in your body, right? Better to get rid of them. But a few of them will hang out forever as memory T cells. And they'll actually go to all of the tissues of your body, poised, ready, already licensed to kill, not like a naive T cell, ready to respond at a moment's notice to start fighting off the virus if you ever get infected again, right? Now, all of this, unfortunately, from the stage of being a naive B cell or a naive T cell, takes time. These things don't happen in the blink of an eye, right? A cell, in order to divide, even to go from one cell to two, it needs to accumulate enough nutrients to grow and double in size. It has to actually go through the process of splitting. It has to produce antibodies. You need the ribosomes to actually have time to build them. And so to go from these naive cells to this full-fledged response where there's antibodies being made, there's memory B cells being pumped out, there's T cells actually killing the virus, takes 7 to 10 days in the first time you ever encountered a new virus, right? That's not super great, right? Because by definition, that means the virus has 7 to 10 days to go relatively unchecked, right? You have other aspects of your immune response that we don't have time to focus on that are doing their best, trying to hold off the virus in the meantime. But that's really where a lot of the drama of how your infection is going to unfold takes place, is in these first 7 days when you really don't have anything to fight with. And then you'll get something eventually, but it might be too late. The virus may already have established too much of a hold, right? What's really nice about these memory cells, right, even if these antibodies go away, let's say the antibodies do go away, and you didn't successfully generate a lot of long-lived plasma cells that kept your antibody levels high for the rest of your life. Your antibodies went away, and so you could get infected. The virus could get into your cells because there weren't enough antibodies around, right? What happens then? These memory cells re-engage and basically do the same response, but much faster. So instead of 7 to 10 days, we're talking 1 or 2 days, 3 days, before you can start to see really clear signs of the activity of these memory cells. So it's much faster. These memory B cells become plasmablasts. Now they make antibodies that don't just make this original antibody, but they make all of those different versions of the antibodies that we talked about before. So if you got infected with Omicron, you have memory B cells that have mutated to be better at fighting off Omicron than these first antibodies that you made, right? And these T cells immediately can start killing infected cells and proliferate and make more copies of themselves, right? So that's the beauty of making memory cells, is that the problem is, because we generate all of these cells randomly, we have billions of them. Your body has no idea which ones are useful and which ones aren't, right? Only a very small percentage of them are useful. Once you know that one is useful, right, you keep versions of it around as memory cells that can respond really quickly because you know this is a virus you might actually see again. You saw it once, you very well might see it again. And so that's like the beauty of a memory response, is that it can happen much faster and make the disease less severe than it was the first time you got it, because the virus has less time to replicate and spread without you exerting any control. So how do B cells and T cells actually receive that signal to become mature? As I mentioned, it's exposure to viral proteins, right? So there's 2 routes in reality now that we can take, right? We all had, at the start of the pandemic, B cells and T cells, ready to fight off SARS-CoV-2, but they were naive, and they didn't know that they were useful, right? And so one way, and the only way we had available to us for the first year of the pandemic, was to get the virus, right? But it's a pretty nasty way to get exposed, right? All of this is happening. The virus is infecting your cells. It's making a ton of new viruses that are infecting other cells, that are spreading to other people who you love. And for 7 days or so, it does this relatively unchecked, right? And that's extremely dangerous. And that's why so many people, unfortunately, have suffered the pains of severe COVID and even died from the disease. Because no one had any immunity to this before. Everyone who gets the virus before the vaccine, right, has to go through this process of letting the virus do whatever it wants for 7 to 10 days. None of us got it when we were a kid. We don't have any memory cells yet. Now, thank God, we have an alternative, which is to show these B cells and T cells the viral proteins that they are meant to recognize, right? To educate them, you are an important cell. You make antibodies I want. You're a T cell that I want you to kill things when you see what you see, right? And this is much simpler, I think. So the mRNA vaccines work like this, right? They have a little piece of mRNA inside a lipid nanoparticle. The lipid nanoparticle fuses with the cell membrane and puts that little piece of mRNA into the cell. As we know, as soon as an mRNA shows up in a cell, the ribosomes will use it as instructions to build proteins. And in this case, the proteins they build are the spike protein on the outside of the coronavirus. That protein can then show up on the cell surface for B cells to recognize, and it can be chopped up and shown as that little signal for T cells to recognize. And those naive B cells and naive T cells that you've had your whole life waiting to see SARS-CoV-2 spike protein can see that on the surface of this cell that really is still doing everything it's supposed to do. It's received very little perturbation from its normal life. It can still do everything it's meant to do. All it's doing is producing a little bit of spike protein. And it's not, this RNA can't replicate. So it's not like the virus where it's spreading. It gets into this cell, it makes spike proteins, and then it goes away, right? Our photocopies aren't very long lasting, so the RNA doesn't hang around for all that long. These B cells and T cells that were naive can proliferate, multiply, become memory cells. They can make antibodies so that if the virus shows up and tries to get into your cell, it can be neutralized. Which I think is really a beautiful thing. And so you can see the difference between getting exposed through infection or through vaccination, right? This is just how things are, right? They're both ways to educate your immune cells to start producing antibodies and become memory cells that can protect you. One comes with this really intense risk, and one is much safer, I think. But ultimately, they converge on the same principles. And I think the reason I started with what I did about the flow of information in the cell, from DNA to RNA to proteins, is that also, I think there's a lot of mystique around the mRNA vaccine, right? Because it's a new vaccine. We've never had anything like it before. And that's true. It's a really remarkable technology. But it's not a huge paradigm shift. Basically, all vaccines converge on the same idea, right? Which is that you need to produce some version of the viral protein so that those naive B cells and T cells can become activated, right? That's what all vaccines hinge on, right? In a safe way, we're going to show the parts of the virus that we want our immune system to recognize to our body. And you can imagine there are lots of ways to get there, right? Like you could put in the DNA, right, which could be photocopied into RNA and made into proteins. You can put in the RNA. You can put in the protein itself, right? So you can make a vaccine out of the whole spike protein. You can make a vaccine out of just one part of the spike protein that we think is especially important. You can make vaccines out of the inactivated version of the actual virus if you wanted to, right? So you could take a SARS-CoV-2 virus, inactivate it chemically, and use that to show the proteins. But at the end of the day, all of these things, from traditional vaccines to the more new age vaccines like using viral vectors, the mRNA, they all converge on the same idea. How do we get the viral proteins expressed in a safe way in our body? It's as simple as that. So I hope that a little bit demystifies some of what the mRNA vaccine is, and also lets you see that it's a really cool technology. With that lipid nanoparticle, you could put in any piece of RNA that you wanted to provide instructions to any cell to produce something, which I think has a lot of potential for biomedicine, as a little aside. So again, you really have two different ways for getting this spike protein expressed in some cells so it can be recognized by your immune system to activate it. And that, I think at the end of the day, the decision is as simple as that. How do we want our naive cells to be stimulated? So now I want to use the last bit of the talk, because I think everyone is curious about this point, to talk about what is actually the current understanding of immunity to this virus. How is it working? Why do we need a third dose of the vaccine? Are we going to need a fourth dose of the vaccine? Does the vaccine work against Omicron? All of these questions. So I'm going to take a drink of water. And we'll dive in. So this is what I study. I work on understanding how immunity develops to this virus after infection and vaccination. So this is just a little reminder of the things we've covered. I think you all know this perfectly by now, so you can explain it to your friends and family, I hope. I hope I'm not joking. I hope it's actually true, that you have a pretty good sense of how this works, right? So the things we're looking for: can we make antibodies, right? Do those antibodies last? Like, do we see evidence of long-lived plasma cells, or are they just going away right away, that we're only really getting these short-lived antibody-producing cells? Do we find memory B cells that are going to be able to generate new antibodies faster if we need them? And do we see memory T cells that will be able to help us fight off the infected cells if we ever were to get infected, if our antibodies fail? Which I think we all unfortunately know, the spoiler alert, that at least some people are getting infected after vaccination. So this is from our paper we published last year, where we followed people getting these mRNA vaccines from before they ever got the vaccine, after both doses, and we've been following them now for over a year, but at the time out to 6 months. And in blue we have people who never got COVID before they got the vaccine, and in red we had people who had COVID before they got the vaccine. So you can see if you had COVID before, you have some level of antibodies from getting infected, right? Some residual levels of antibodies left, and if you never had COVID, of course, you don't have any antibodies, because it's a new virus. Where would you get them from? If you get the first dose of the vaccine, if you had COVID before, right, what are we thinking? If you had COVID before, you have some amount of antibodies left, but you also have these memory B cells, right, that can very quickly produce a lot of antibodies, because they're memory cells. They're better than naive cells, right? And you see that with a single dose of the vaccine, these people produce a ton of antibodies, right, and reach really high levels. And the second dose can't really get them any higher. Like, they've basically reached a maximum potential, right? If you're naive, right, that first dose, you produce some antibodies, which is nice, but the second dose clearly makes you produce more. It's still not as good as a full-on memory response, right? Because these people are getting to have a memory response to the vaccine. This is still an initial primary response, but this is a good level of antibodies. And this was the level of antibodies that gave 95% clinical efficacy against SARS-CoV-2 infection, right, when the clinical trials were done, which is remarkable. If we could keep antibodies here forever, we would probably have something in the realm of 95% efficacy forever, which would be amazing. Unfortunately, what we saw, and what many other labs have seen too, is that over time those antibodies do decline, right? And it's not that you go from being immune to being susceptible, but you gradually become more susceptible to the virus. And if you have bad luck, right, like with even this level of antibodies, you can be one of the 5%, right? And if you have, good luck, even as your antibodies go lower, you might get exposed and not get sick. Those are normal things. It's normal that 2 people get exposed and 1 gets sick and 1 doesn't, right? But it's all, you become more susceptible. There's a greater chance that you might get infected as these antibodies start to fall off. We also looked at the memory B cells. This is extremely positive news. So the mRNA vaccines are awesome at making memory B cells. So you see after infection, you have a lot of memory B cells. If you give the vaccine, you make a bunch more, and then they kind of return back to where they were before. What that means is that you already had a healthy level of memory B cells from the infection, which is good, right? And that's the reason you were able to make so much antibody after that first dose, is that you had this many memory B cells hanging around, ready to produce antibody, right? If you never had COVID before, of course, no memory B cells. After the 2 doses, they go up, and then they actually stay very strong or even increase over time. So you have an extremely stable level of memory B cells for months following vaccination with no indication that they're going anywhere, which is really great. It means probably these memory B cells will be there for the rest of your life. If you ever get another dose of the vaccine, you'll have this kind of response and make a ton of antibodies. If you ever get exposed to the virus, you'll have that kind of response and make a ton of antibodies, which is a great thing. And I'll just say quickly, we saw the exact same thing with the T cells. So they don't keep going up, but they do a very classical T cell response. You make a bunch of them, and then they come down, and then they establish themselves at a certain level of memory, T cell memory, that actually looks really good, and we've seen can get re-engaged when you get exposed again to the virus. So hopefully, like you can see, going back to this picture, at least initially, we generate a lot of antibodies, we generate a lot of memory B cells, and we generate a good number of memory T cells with mRNA vaccines, which is great, right? It means the immune system is working how it's supposed to, and it should be able to give us some protection, right? Unfortunately, we did see that the antibodies were coming down, and I think that makes us all curious, like, can a third dose help those antibodies stay high again? And so this is unpublished data from our lab, so you get a treat. You're the first people to ever see it, but hopefully published soon. So here's those same people. So the higher you are on this scale, the more antibodies you have. And these are specifically the neutralizing antibodies for the original strain of the virus that the vaccine is meant to target. So you can see after the first dose, you don't actually have that many. This is why it's really important that we did a 2-dose regimen. After the second dose, you have quite a bit, if you were someone who never had COVID before, and those start to come down. What's interesting is that looking longer than 6 months, we did see them stabilize, right? This suggests that there probably are at least some of those long-lived plasma cells continuing to produce antibodies. The problem is that they didn't keep the antibodies up at this level here, where we saw 95% clinical efficacy, right? If you get a third dose, wow, those antibodies go through the roof. And it actually looks, unsurprisingly, because of what we now know about memory B cells, it looks exactly like what we saw in the people who had COVID when they got their first dose, right? You produce this memory response from your memory B cells, you make a ton of antibodies, and you reach a really high level of protection after the third dose that actually you never had after the second dose even. So this, I think, to me, it shows a lot of value to the third dose. The little X's are people who got an infection before they got their third dose, and you can see that that achieves the same result, more or less. If you get a third dose and you had COVID before, you do get a boost. It might be a little bit higher than you were before, but it's fairly comparable. There probably is a sort of maximum on how much antibody you can really have. These were already super high levels. What's really encouraging, and that I think is one of the big questions we still have to answer now, is that these people started to plateau at a pretty high level of antibodies. So it's possible that this memory response generated more of those long-lived plasma cells, right, that can keep producing antibodies for a longer amount of time to keep your antibody levels high longer, right? And if it's true that this recall response generates those kinds of cells, it's possible that we'll see the same thing here. But we don't know. We don't have the data yet. It's too soon after we started giving third doses. So we'll have to wait and see. But I think there's at least a slight reason for some hope, or at least curiosity on my part, on whether we'll see the antibodies just always decline forever, or if we'll see them start to level off at gradually higher and higher levels, which would be great. It would mean we're more protected, less likely to get COVID over and over for the rest of our life. Unfortunately, my clicker stopped working. It's not what I was going to say after unfortunately, but Matteo, can you advance the slide, maybe? Thanks. So, unfortunately, Omicron happened, right? So here's a picture of the SARS-CoV-2 spike protein with some of the mutations that were found in the Delta variant on the left versus the Omicron variant on the right. And you can see why this is a problem, right? If the virus is changing a lot, right, the antibodies that used to stick to this part, well, all of that part has changed, right? Which is a problem, because those antibodies might not work anymore. Now, there's still a lot of it that hasn't changed that much, and those antibodies can still work, right? But some of the antibodies aren't going to work as well as they used to. So here is just another group's data, because theirs is a little nicer than ours, admittedly. So this is people 1 month after the second vaccine dose and 5 months after the second vaccine dose. So they see those antibodies declining again, right? Each dot is a person. So you see there's some variability, but there's a little bit of decline over time after the second dose. And after the third dose, those antibodies go up. Now these are antibodies against the original strain. If we advance the slide, we see with Omicron, even after the second dose, when you had very high levels of antibodies that work against Omicron, or that work against the original strain, you have pretty low levels of antibodies that work against Omicron, right? That's because after the first dose, you're only really activating naive B cells, right? The B cells that produce an antibody that's good at recognizing the original strain. But if you remember, when you make memory B cells, they diversify a little bit, right? Some of them might randomly diversify to be better at recognizing Omicron. And in fact, when you get a third dose and you reactivate those memory B cells, you get better antibodies against Omicron. They're still not as good as the ones, they're not as high as the ones against the original virus, but they're there. So I think this is a really strong piece of evidence in support of getting a third dose of the vaccine, specifically in the world now where Omicron is present. That the vaccine does work against Omicron, but you probably need at least 3 doses for it to work. And so with that, I just want to leave us with the big picture that it works now. This is what immunity looks like, right? I hope we've learned a lot. I hope we've learned how we make such a tremendous diversity of naive B and T cells, how we select the cells that we want, how vaccines can help us to do that in a safe way, and what the landscape is for immunity against this virus that we've been talking about ad nauseum for 2 years, to hopefully help you understand why are they telling me to get a third booster, or why are these other people telling me not to, and what's really in your best interests, and hopefully you'll have an understanding of immunology that you can carry with you. So God forbid there's another pandemic, like we can understand what's actually happening in our bodies when we experience a virus. And I just want to say that there are a ton of unanswered questions. I hinted at one of them with where are the antibodies going to level off in the end. That's something we really don't know. And how is Omicron going to change the game in terms of long-term immunity, among a million other questions. So science is really a process. We've learned a little bit. I think we've learned a lot. We've had some big successes, and a ton more to learn. And so new information will always be coming out, and hopefully now you'll be better equipped to process it and understand what it is. So thank you so much. Thanks to the New York County.